Good afternoon, everybody. Like Mutab said, my project is entitled Investigating the Role of Mucosal Immunity in Regulation in Mendelian Susceptibility to Mycobacterial Disease, a case study. Specific patient in the primary immunodeficiency disorders genetics research group study cohort presented with a primary immunodeficiency PID for short and unique clinical diagnosis that involved chronic diarrhea, salmonella infection, as well as Clostridium difficile. In addition, the patient was unable to follow a proper eating regime from the age of 2 weeks. Further investigations were required to make a definitive diagnosis. Whole genome sequencing and functional work was conducted previously to determine whether there is an underlying explanation on both genetic and cellular level, which aids in proper prognosis of the patient prior to the stem cell transplant. Whole genome sequencing revealed 2 gene variants, namely IL-12RB1 and FOXP3, to be potentially associated with this unique clinical phenotype observed in this patient. IL-12RB1 is well associated with a PID known as Mendelian Susceptibility to Mycobacterial Disease, MSMD for short. However, the patient never developed mycobacterial infection even following BCG vaccination, but suffered from invasive salmonellosis at 1 year. Salmonella infection, on the other hand, is also well associated with MSMD, therefore classifying this particular PID as MSMD in the absence of mycobacterial infection. Additionally, the functional work conducted prior to the stem cell transplant showcased a decrease in IL-12RB1 receptor expression on several lymphocytes. The functional work focused on the IL-12 interferon gamma pathway. However, the patient had a stem cell transplant, but some of the complex clinical features still remain, specifically those relating to the gastrointestinal tract, GIT for short. Based on the entirety of information supplied from the whole genome sequencing, functional work, and clinical reports, there is a clear indication that the GIT of this patient should be studied more intensely. The GIT contains a large microbial community and is therefore known that gut microbial settlement in the GIT is essential for the formation and establishment of the immune system. The present study will investigate the mechanisms that disrupt intestinal homeostasis and influences mucosal immune regulation. We hypothesize that dysregulation in mucosal immunity and dysbiosis of the gut microbiota is contributory to the clinical phenotype observed. Therefore, specific aims for this particular study is firstly to investigate the microbial immunity composition of the gut microbiota before and after the stem cell transplant. Secondly, to compare the metabolite profiles of the gut microbiota before and after stem cell transplant. And lastly, to describe IL-12RB1 expression and IL-12 signaling after transplant. The main goal of this particular study is to unravel functional interactions between host immunity and the gut microbiota. We anticipate to identify differences in the gut microbiota profile as well as the gut metabolic profile pre and after the stem cell transplant. And lastly, we hope to elucidate the mechanisms of microbiota modulated metabolite immune regulation in the specific patient. I would like to acknowledge the following individuals and thank you all for listening.
Thank you very much, Brandon. That sounds like a very interesting project. Are there any questions from the audience? Okay, while we wait for any questions anyone might have, I just want to know, what is your sample size for this? I don't know if I missed that. You might have said it.
Okay, so my sample size, because this is a specific it's a specific patient that presented with this unique clinical phenotype. The idea is to firstly establish and describe the metabolic and gut microbiota profile in this patient with this particular PID, as it has not been done before. And hopefully, if the study goes further, we hope to recruit more patients. But this is one of the most unique clinical presentations seen in the PID cohort.
Okay, thank you. We have a question from Penelope. She asks, you mentioned BCG vaccination. Has it been linked to gut dysbiosis in the literature?
So far in literature, I haven't came across BCG vaccination, but we included that this particular patient did receive BCG vaccination, as we know that MSMD deals with mycobacterial disease or susceptibility to mycobacterial disease, but however, this patient never had any episodes of MTB. But I will look further into infections that deals with BCG, but BCG is also just a vaccine to protect the infants and children against developing MTB.
Okay, thank you. And then a question from Natasha. Would it be possible to compare this patient to children of a similar age?
So using controls in microbiome work is quite challenging, because however, we don't know what parameters to include exactly, as you would want a control of a similar age, someone who grew up in a similar environment, as we know that the environment also plays a large role in the establishment of the gut microbiota. But what is normal for a gut microbiota profile? Because I believe everyone has a different gut microbiota profile, but however, these differences observed between those that develop a certain disease or infection and those that are not prone to develop that.
Mm-hmm. Okay, thank you. I'm going to take one more question. From George, is it difficult to evaluate microbiome profiles in South African patients without a larger, a normal reference base, and how could we get around that?
So to answer your question briefly, this is because Pigeon primarily focuses on finding novel gene variants associated with PIDs. Only function work is conducted in terms of immunological responses, but with this study, it's one of the first actually that we are interrogating the gut microbiota. But in future studies, it would be good to see or have a larger study cohort to see how South African patients, especially children, are doing.
Okay, thank you so much, Brandon.